that. This is the pointer, right?
I am glad to introduce the next speaker, Dr. Malini Laloraya, a senior scientist at the Female Reproduction and Metabolic Syndromes Laboratory at Rajiv Gandhi Center for Biotechnology, RGCB. Madam already chaired a session and has been introduced, but still, till the slides are being arranged, I'll quickly introduce again. She's a visiting faculty at the Rockefeller University, University of Virginia, University of Florida. She is the proud recipient of the Govind Rajulu Medal Award, the GP Talwar Gold Medal, the RAIN Visiting Fellowship, and several other accolades. Over to you, Madam. Thank you.
Thank you, Chairs, for the introduction. At the outset, I would like to thank the organizers for giving me an opportunity to talk to all of you. Well, this time I am not going to talk on implantation. So, since everybody has been telling from the morning that I work on implantation, I do still work on implantation, but I will talk something on, it's always research is a learning process. So, I have learned a lot in the last 4 or 5 years about circadian rhythm. There are stalwarts of circadian rhythm I know in this audience. So, I think they will excuse me for any errors. Does it move? Yeah, this thing is not working. I have to show here?
No need. You can show there also.
Okay. No, it doesn't. You have some problem in it. There is some problem in this. Okay, I will start here, okay, by the time they fix it. No, you just first connect this. Let me try. Nahi ho raha na? Yeah. Can you put this in here? Go to the first slide. But how will I point then? Get me a laser pointer. इधर से कर देंगे आप इसको यहाँ रख सकते हो नो आई विल डू विच इज़ द नेक्स्ट वन ओके ऑलराइट सो आई विल ट्राई टू डू विद दिस ओके सो वेल एवरीबडी हैज़ टॉक्ड अबाउट reproduction, infertility, both male and female, so I will be quick on these. So, the female factor infertility is what interests me. And out of the female factor infertility, PCOS is one of the major issues which we are dealing up. This is not good. Okay, now it is working. So, since everybody talked about embryo implantation, I thought I should show one figure on embryo implantation because this is most recent. We have developed spheroids and an in vitro assay system for implantation. We are also into developing organoids from infertility patients facing recurrent implantation failure. But today I will just talk about polycystic ovarian syndrome very quickly, and this is called as a thief of womanhood. And for the younger students, for introduction, this was discovered in 1935 by Irwinstein and Michael Leventhal, and they had just 7 patients that time, 7 women which had varied manifestation with acne, hirsutism, obesity and bilateral enlarged cysts. And now what is known is that it is a triad feature. They have oligo-ovulation or anovulation, hyperandrogenism, and which leads to acne, hirsutism and alopecia. Hirsutism means male pattern hair growth, and polycystic ovaries are there. So, this is a normal ovary and this is a polycystic one where ovulation does not happen. So, you have lot of peripheral cysts there. And so, this is the classical triad features of polycystic ovarian syndrome, and there were several large meetings to decide on how to diagnose PCOS, and these societies have given diagnosis. So, you have the NIH criteria, Rotterdam as well as androgen axis. I am not going to read it to save time, but what is largely followed is the Rotterdam criteria, which is 2 out of these 3, that is you should have either hyperandrogenism oligoanovulation or multiple cysts. These are followed, and of late actually there is a newer classification in this which subtypes the PCOS patients. And if you look at the global prevalence, PCOS is on the increase, and there is a difference in with ethnicity, and India comes at the top. And in India especially, there is, this is what is some time back a review. In India there is a alarming increase of PCOS cases which our country needs to worry about. And so one in every 5 adult women could have PCOS. Anyone wanting to have details, younger students can contact me. I am there on all the social media, and email is there. Fortunately, I have a very odd last name, so it is very easy to get me. So, you have genetic and epigenetic factors which regulate, environmental factors, and the major core is that it is a neuroendocrine disorders with high LH and low FSH, where this ratio is more than 2, and that leads to altered steroid production and hyperandrogenemia. Now, along with this, in PCOS you have different other comorbidities, and these are well established. Cardiovascular disorders, diabetes is very well known. Reproductive abnormality, they have more difficulties in conceiving. Implantation rates are very low in these subjects. Overweight and obesity is a known thing. And there was an interesting comorbidity, sleep disorder and depression. And how this interested our group was very interesting. I had a student, Meera Krishna, who worked and did global profiling in 2006 or 8, very early we started. I have not released the data because I learned from one of my earlier labs where I worked as a PDF not to release this data and keep on working on what comes out. Instead of making one paper, you make lots more. So, so she actually showed that time that in PCOS you have problems with immune aspect, and we are the first ones to talk about an immunological problem associated with PCOS, where we showed that PCOS subjects have low T regulatory cells. Now, the other so there are a lot of pathways, and these are there everywhere. So, I do not like to touch some things which is there everywhere. I always like to touch the odd one out. So, we had circadian rhythm which interested me and excited me, and then we did a study literature and looked at circadian gene regulation. So, BMAL knockouts are infertile, clock knockouts have metabolic syndromes, you have ROR beta have male reproduction problems, and these two again correlated with our immune aspect, that is lymphoid organ development and abnormal lymphoid homeostasis, which we are continuing through some ICMR grant currently. So, what is the circadian rhythm? We all talk about it, and this is that our body perceives light and through the suprachiasmatic nucleus, and we have a 24-hour, approximately 24-hour rhythm. You do not have to read what is written, I can just speak in short and do faster. And so, this is the entire clock machinery in us. So, the BMAL and clock bind EBOX of several genes, candidate genes. Out of this, some are autoregulatory loops which are called as the negative loop, which regulate BMAL and clock. So, Rev, Cry, Per, all of these regulate BMAL and clock, and these are the core clock genes. Okay. And then you have circadian clock genes which finally regulate all our body's physiology. Now, 2008, I must tell you the story. My student, when she was doing in this PBMC, when we got the thing, I told let us look at clock genes. So, she said no, but it can't be there, it is not a, it is in the brain. Anyway, we looked, she got it, but she was also very hesitant. So, we left it. 2008 there is a Nature paper, just a small note reporting the existence of peripheral clock genes. We saw it first, but did not report it. So, now it is known that you have these peripheral clock genes, they are there in all our organs, and these are called as the peripheral clock genes, including the immune system. So, what happens is you have a central clock which regulates the peripheral clocks, and then they regulate metabolism and physiology in all the organs. So, as I said, Along with the genetic factors, environmental factors also are key in regulating reproduction, and there is a positive correlation in night shift workers and PCOS. There is also, many people quote foreign work. I found out a very interesting paper of 1969, I do not know who Singh is, but he made a continuous light exposure model to show that these animals develop PCOS features. I am sure Professor Chandana knows who the Singh is. Okay. So, and approximately 80% of our world population is now affected by light polluted skies. So, one is that we are working odd times, the shift workers, and the other is that we are exposed to excessive light, which was not there earlier. So, this is impacting our biology. So, the question was, is circadian rhythm dysregulated in PCOS patients? So, we took blood, because to answer many people who always said you are doing in blood, it might happen in brain and ovaries and all that. Fortunately, nobody was ready to give me brain and blood human beings. So, I had to deal with blood only, and we isolated DNA, RNA and protein. And for the student community, if you are doing human work, you have to follow all the norms. So, you need to get human ethical committee approval, and we did for 101 normal and 101 patients. It was very challenging because it was during COVID times, and I had a very hard working student who could accomplish this, and we had the inclusion criteria and exclusion criteria as laid down by Rotterdam. And if you look at the biochemical characteristics, yeah. So, LH was high in our subjects, testosterone was high. So, the classical features were there, and as I said, you have phenotype A, B, C and D now for PCOS. So, we also analyzed our data based on that. And yes, it was very challenging because for 200 samples we did real time PCR for several genes and very expensive too. So, we could very neatly show that clock genes are down regulated. These are called as violin plots, and they are generated in GraphPad, and BMAL is down regulated as well as NPAS. We could only get blots for clock because in the peripheral network the amount present is very less. So, we could show even that the protein for clock is down, and if you look at the phenotypic analysis, all of them, the phenotype A is most affected. The phenotype A is around 56% in the Indian population, and the negative loop was upregulated. If you look at this, so this is what I am talking about is these PER and CRY, which are the important negative regulators, and these also are affected mainly in the phenotype A. More genes for the negative loop, CRY 1, 2, DES1 and 2, all are upregulated, and therefore, finally, it all ends in a dysregulation in BMAL and clock. So, for CRY also we have the phenotypic analysis in our patients, and finally, the regulators of PER and CRY as well as ROR alpha and beta. So, both are this is upregulated, this is not that significant. So, we believe that CSNK is one of the major problems here, and the inhibitor NR1D is the inhibitor for BMAL, and that is upregulated, and the ROR-A which promotes or stimulates BMAL is downregulated. So, everything finally leads to lowered BMAL and clock in these subjects. And I will skip I think the phenotypic thing. So, the reviewers asked us a very challenging question. If this is circadian related, do it in different time points in women. We could only counsel the women, and they gave samples for 10 a.m., 2 p.m. and 6 p.m. and we did not get the other two time points in the night, but we do definitely see a phase shift in the gene expression for the core genes. And if you look at it, so the lessons learned till now is that expression of core genes is down regulated, negative loop is up regulated. Among the 4 phenotypes in the Indian population, we have 56% of phenotype A, and this is the one which shows highest variation. Now, what is the functional implication? What if the circadian clock is dysregulated? What does it do in us? So, what you see here is an interesting connection. So, elevated androgen levels are the key biology, right, for PCOS. So, you have high testosterone, high DHT, but this is the serum concentration. Okay, DHT is very less in the serum, but the biological activity is highest. So, and low estrogen is concentrations is another primary defect. Human PBMCs interestingly are known to be a site for peripheral metabolism of PCOS, of steroidogenesis, and it is shown that monocytes, lymphocytes, they all have this machinery for peripheral androgen metabolism, and BMAL is another important for steroid synthesis, and loss of BMAL results in reduced testosterone, estradiol as well as progesterone gene production. Star gene also is a direct target of BMAL. Now, this is a figure which I thought I will never have to read. My teachers in MSc made me draw this chart when I was a MSc student, and it came to use. So, so star is involved there. You have 1720 lyase here, and this is the pathway, which is the blue steps are in the peripheral tissue, the yellow is in the granulosa cells. Okay. So, I will like you to focus on this 5 alpha reductase and the aromatase. So, if testosterone accumulation has to occur, number one, this pathway has to be there because that is the core, but then if the pathway should go this way and not this way, that will be the theoretical aspect. So, we looked at this aspect and we first checked their expression levels. So, star was high, CYP17 was high. I will not talk about the phenotypic right now to save time. How much time do I have?
6 minutes.
Okay. Is there some water? And then, as I said, we need to worry about this pathway. So, both aromatase and alpha reduction is coming again. Just a minute. Yeah. So, this is the aromatase. So, interestingly, aromatase was down in PCOS subjects. It is a very core endocrinology disorder, but nobody had looked at these genes. And SRD5, A1 and A2, which make testosterone and DHT, they were high. And so, we also did predictions by contra V3, and we could show that CYP17 is a direct BMAL target. So, we did CHIP, and here we show that clock BMAL can bind to the E-box on CYP17. And we did CHIP also and could show that CYP17 is a direct clock BMAL and BMAL target. And then what we designed the experiment to show the functional relevance is, if you silence clock and BMAL, what happens to testosterone to DHT conversion? And for this, so first we silenced BMAL and clock and checked that it is efficacious. And then we saw that if you silence BMAL, clock and clock BMAL together, you see a down regulation in CYP17. So, your initial steroids which are required for the biosynthetic program are not there. CYP19 is down regulated, that means you are not going to make estrogen. But SRD5A and 5-2, both the reductases are very high when you silence clock and BMAL. That would mean that the system is being pushed to make more dihydrotestosterone. And this was a little difficult task because we had to do upon silencing and in culture, and we wanted to measure estradiol and dihydrotestosterone levels. So, when you silence these molecules, the testosterone levels is there, but estradiol levels comes down and dihydrotestosterone goes up. So, this finally proves that silencing of the gene not only is affecting the RNA expression, but it is also affecting the final end result, that is the steroid hormone. So, I will skip these slides for reading, and so what we suggest is clock-B-mal is involved in directly regulating testosterone and DHT conversion in the periphery. And so, one of the reviewers asked, and we also wanted to do it, what about ovaries? So, we can't get it from the human beings. So, way out is we made the animal model, the androgen excess model, and assessed ovarian clock, and I will skip this a bit, and we did all the classical things, the estrocycle measurements, the ovary, they had lot of peripheral things, body weight, there was a change, but most importantly, obviously we are injecting testosterone, so testosterone and DHEA is high, LH became high, LH FSH ratio was high. So, the melatonin levels were high, and the core clock genes, they all showed a phase shift, whether it was B-mal, clock or PER2, as well as there was loss of circadian entrainment in the peripheral blood of mouse. So, that was another very daunting task. So, we took out peripheral blood from mouse at different hours, Rydberg hours, and we could show that there is a phase shift in the circadian clock genes. So, the rhythmicity is lost, and we measured the GTA cycle and looked at the acrophase, and we found a significant shift in these animals. So, finally, we have given the concept that in PCOS you have altered peripheral clock, which leads to hyperandrogenism, and this is also proved by this model. I am not showing data of this right now, I might not be able to speak longer. So, interestingly, I am very happy that our paper got published in June in Human Reproduction on this work, and let us just skip it because I have told, this is important for the younger generation. So, I am sure all of you, when your parents tell to sleep, you still look at your mobile. I am also educated that you do not look at Facebook, you do Instagram. So, all of you do that, but keep in mind that it would affect your reproductive health. So, hopefully, I will encourage you people to sleep on time, and it also necessitates policy development for our government for regulating night shift work, because see, some of you students might tell that we all work in the night, but we do not work every day, and our work demands. If I have to study implantation, I work in the night. IT work can be done in the daytime also. We do not have to be that much this thing that if the foreigners tell us to work in night, we are working for them, they do not work in the night for us. So, we might have a country which is affected by PCOS, diabetes, all of them. So, this work was started by Meera, the microarray only was done by Meera, and that was a starting point, and we had very good discussions with Meera, and I have two wonderful clinicians as collaborators, but I would give full credit to Betsy, who did all the work, mouse work, human work, and in very difficult times during COVID, all the mouse work in the night was done when there was lockdown. So, acknowledgement, well, all this work was funded by DBT, both my intramural grants as well as extramural. DBT has been very nice to me, and I have never had dearth of funding. Patients and staff of all the hospitals, obviously RGCB facilities were there. Thank you so much.
Thank you, madam, for a wonderful talk. I am sure there might be a few questions. Yes, over there.
Many are there, yes. Okay.
It's really great. In fact, slightly different, which we usually expect.
Okay.
So, it's just a a clarification. As per your mouse studies, it looks like a consequence, but as per your human study, it looks like a cause. So, what do you say? Is it
It's a
cause or the
chicken egg
consequence?
question, but I think it is a consequence because we have also now made the light model of mouse, and that also is showing similar data. I did not want to show unpublished data because this is being video recorded. I had it, I removed it, all of it. So, maybe next time I will talk on implantation though, but still. I think it is a cause of PCOS because if you see the sudden increase in India, think about it, all our kids are working in the night because they have to match time with the US people. And this is not just about PCOS, there are other alarming diseases which I discussed with some clinicians which have never come up here and now they are being expressed.
Yes, Dr. Kanakraj.
Somebody else was there, yeah. There is one student also there, right? Yeah.
It's a wonderful presentation, madam.
Thank you.
I'm going to ask a funny question. Whether the mouse or rats are right model to work at the nocturnal?
It's not the right model definitely, but that's the available model. Sometimes you are bound by, like people ask me in the human, is it why you not doing in the ovary? So or say you want to look at the brain. I said if anybody is ready to give me, I will be happy to do it. So if you find people, please, I will encourage.
Sorry, I don't have any ovary.
At the back here.
Yeah, there is some student on the back. Can we first?
Very interesting talk, ma'am. Ma'am, I have two questions. First is you have related the circadian rhythm with the PCOS. Is there any study which relate the same thing in the males also? Right. And the second question is you have mentioned the peripheral cloak present in so many organs and tissues. So is there any correlation between the PCOS patient with any other disease which is common in both, as you have dealt with the clinical samples?
Which is common in both?
Both, like in case of PCOS, any other disease related to
Circadian rhythm.
Yeah.
Okay. So answering your first question first. So it's thought that any disease is not classified as a disease if it doesn't have the male counterpart also. So now there is also a male counterpart. So men who bald earlier, very early, okay, are thought to be, that is one of the criteria for checking whether they have the male phenotype of PCOS. So balding occurs because you have high, that time you have very high dihydrotestosterone. PCOS patients also have high DHT. So that is thought to be there. Your question was also asked to me by many Air India, the people who service the stewards, because women have problem, they understand, but they discuss lot of problems which they have, but no study is there.
Madam, I would like to interrupt. There is a study on flight stewards that has been done.
Now it is done, right?
And in the transcontinental flights,
Yes.
and depression has been shown in them.
Yeah,
in the male
depression
couple.
is there, but not the any other male reproduction related
No study
stuff.
yet.
Yeah, no studies there. These talks I used to have before COVID when Air India we used to take to fly and they used to chat with
Dr.
us.
Asumanjo, a quick question because we
Yes.
are already out of time. Last question, please.
You can also talk to me later if you want. Go ahead.
No, actually, it's a nice one. So the thing is that what we have shown about the phase shifting in female
Yes.
clock and your melatonin data, what is showing. So I think because if you've seen zebrafish, it has been elaborated that if you expose to them to the artificial light for a longer period of time, there is phase shifting and there is ovarian tumorigenesis is there, which is very similar to the PCOS stage. So but your melatonin data, what is the shifting of melatonin in case of, it is not showing actual shifting in the
Yes.
mammalian clock. So can you?
So we are actually working on that angle because in the human we did not do that time the melatonin assays because others have already done. Melatonin has been in PCOS subjects, they have checked in urine, in blood and ovaries.
Sorry to interrupt, madam.
Yeah,
We are
so we will
already out
discuss,
of
that is
time.
what I am telling, we will discuss later.
Even I had a related question, but I'll ask
We
that
will
later.
discuss later.
Thank
I have a problem after
That was
COVID.
clearly a
I
very
have
engaging
gasping.
talk. Thank you, Dr. Laloraya.
Yeah, this is
I
the first
request
time I
Dr.
give a talk on
Devkar to present a memento to Dr. Malini.
So after this, there are a couple of presentations by the sponsors. The first one by Biorad. They can.
So now we'll have some quick presentations by representatives from Biorad and Eurofins. I first call upon representatives from Biorad.
Hi. So at the outset, I would like to thank the